Hello, everyone, and welcome to another exciting roundtable. This is part 2 of our Venus flytrap breeding roundtable, and we're joined by some experts from around the world. And before I do that, you'll notice that I'm wearing a Nepenthes bicalcarata t-shirt. And if you want to support the ICPS education or conservation initiatives, you can go to the link below, and anywhere in the world you can order a t-shirt, a tote, a mug, and with this super cool item on it, or you can get a Utricularia, or you can get Drosera, and you will be supporting the ICPS. All right, so with that, I'm going to have the panel introduce themselves, and then we have some questions from social media that I will address. So let's start with Craig. Can you introduce yourself?
Okay, my name is Craig Heath. I'm the owner of Crazy Craig's Carnivorous Plants. I'm in the U.S. I live in Lorton, Virginia on the East Coast.
I'm Valerio from Diflora, Italy. I live in the north of Italy, and we are a tissue culture lab that is specialized on production of every type of carnivorous plants. What about you, Matze?
Hi guys, I'm Matze here from Green Jaws from Germany, and we have a company for carnivorous plants, of course, and mainly specialized on Sarracenia, Flytraps, Cephalotus, and also doing with some tropical stuff like Nepenthes. But our main thing, I would say, is Sarracenia and Flytrap breeding. And I'm very happy to join the meeting today and talk about flytrap breeding. And I hope you guys can take some very good informations out of it. And yeah, I'm very looking forward to it.
All right, very good. So Matze, we'll start with you. Can you name a couple of kind of famous cultivars or types of Venus flytraps or named Venus flytraps that you've created, and maybe talk a little bit about how you choose the parents?
Okay, yes, sure. So I think a highly recommended plant is the Hellcat, the GJ Hellcat. So every own clone from us has a GJ in the name, so everyone knows it's from Green Jaws. And the Hellcat, for example, is a very nice clone. It goes a little bit into direction from Jaws Smiley Alien, but it's a little also different. And this is a selection we got randomly out from seedlings. So let's say like this, when we do our flytrap breeding, we have different goals. So for one goal is to find very big traps. So we want to create very giant forms with 5-6 centimeters. One of my dreams is to have a flytrap with like 10-centimeter traps. I think we have to work on it for the next 20-30 years. You know, it's always a big selection that you have to choose your biggest plants and you cross them with the biggest plants to create more bigger traps then. So every generation, maybe you get only 1-2 millimeter bigger traps. And if we are talking about the big traps, we have, for example, the GJ Giant Form or the GJ Goliath, which do every year constantly traps with 5 centimeter plus. So these are really giant forms, and I also cross them every year to see if we can get more bigger traps. And so this is one goal of our breeding, that we find very big traps. The other thing is that we want to find very crazy looking fly traps like the Hellcat, or we have also a lot of different selections of pom-pom forms like the GJ Supernova, GJ Coralle, GJ Explosion is also a very nice one. And these are the spontaneous mutations. And so we cannot always say what are the parents of it, because we have like a breeding system where we cross different clones to get a new variation out of it, or we hope to get a new variation out of it. And then we have randomly pollinated fly traps. And we have a big collection with over 300 different fly traps. and they all standing next together. So yeah, they're all mixed also. And from these seeds, the most interesting plants coming out. And yeah, then the third breeding goal is that we want to make some red variations of the forms we already have. For example, the funnel trap. I'm sure most of it know the funnel trap or the cup trap, and we try to get a red variation of it. So a red cup trap is also existing already, but a red funnel trap, for example, it doesn't exist. So every year I try to cross the funnel trap with red forms, but it's very hard to get a red color on the trap, and which looks like a funnel then, because the funnel is not how to say that it's it goes from generation to generation. So it's you know what I
Maybe
mean,
like
Kenny, right?
recessive, like a recessive trait.
Yeah. It's not dominant, it's recessive. So yeah, but I got some fly traps out with stable funnels. So when I propagate them by seeds, they always produce the funnels then, and this is my basic now to try to put a red color inside. So these are our three goals for breeding: giant traps, crazy traps, and red variations.
All right, very good. So Valerio, can you do the same question? Can you name some of your more well-known cultivars, and how do you choose the parents?
Sure. I am very glad that Matze introduced such a very interesting matters to discuss together, because our breeding programs is focused in a very similar way that the one of Green Joe, but with very different technique of seed, not of seed production, but also of genotype generation and selection. For example, our most well-known cultivar is the Carbonia ardenti. That is a very nice selection that it has been generated by crossing between microdent and cup trap. So very, very simple fly traps, but that cross has generated the Venus flytrap called Periscope, that is a registered cultivar too. But the most interesting family of Venus flytrap derived from Periscope has derived from in vitro mutation. So no traditional breeding, but casual in vitro mutation of the Periscope genetic line. So a single mutation of one plant of in vitro cultured Periscope has generated the Carbonia ardenti. That is a Venus flytrap very similar to microdent, but with a deep red coloration inside the trap and a huge amount of trigger hairs on each lobe of the plant. So now the traditional 3 trigger hairs for each part, for each lobe of the trap, but a huge amount of trigger hairs. Then we have had also the red Periscope, that is all red selection of our Periscope, and the Fiamma. Fiamma is a Venus flytrap characterized by the same feature of Carbonia ardenti, but with longer teeth. So while Carbonia ardenti have microdent-like teeth, so almost no teeth, the Fiamma has very, very long teeth with the same amount of trigger hairs as main feature. So coming back on our main way of breeding and selecting Venus flytrap, we have the traditional way, because we have a huge amount of Venus flytrap cultivar too, as Matze, and we breed them also trying to remove anthers and the pollen from some flower to select only the stigma and the female part, so to have quite a nice control on pollination. So we start from seeds and we try to select, after the in vitro germination, plants inside the tissue culture vessel with crazy traps, because it's the only feature we can select as a young plant already in the vessel. While, for example, for the giant feature, we should wait for a bigger plants in the pot, so in the standard way of growing in the greenhouse. And this is one of our way of producing new cultivars. So doing breedings and germinating both in vitro and in standard growing medium, and selecting in vitro for the crazy plants, while in pot for the giant ones. And then we see that using a very light amount of tissue culture hormones, we are able to produce a callus that is in vitro. A callus is a mass of cells that can generate one by one a single different plants. So in that case, mutation could be quite rare, but it happens. And so sometimes the all red mutation that, for example, Matze was searching in his breeding is something that happens quite a nice amount of time. So to select the all red mutation of a cultivar, and also some very rare mutation, like, for example, the one that has generated Carbonia ardenti. So to summarize everything, we use traditional breeding to let the seed germinating in vitro, and at the same time we use callus production for plant regeneration to select the single cell mutation that is able to produce horticultural interesting plants. And then, of course, we evaluate them after acclimatization and more or less on 2 to 3 years of growth in the pot. So this is the way, the main way in which we create new plants, and our aim is more or less the same of all of us. So searching for the craziest traps, but also for large cultivar. As a tissue culture lab, maybe we are more focused on the feature and the plant character that is quite quick to recognize also in the small plants. This is something that is for sure real and true for us.
All right. Before we go to Craig, Matze, are you growing with the assistance of tissue culture, or are you seeding them
No.
in seed or in
We
soil?
do everything. Yeah, we do everything in the old way.
All right. So Craig is enjoying retirement, and he's playing around with tissue culture. So Craig, same question. How are you choosing the parents, and can you name some of the more well-known cultivars you've created?
Okay. Most of my plants that I do, I've been doing tissue culture for like 10 years now. I usually get my seeds from a guy named Stephen Wang. He lives in California, and he usually sends me some seeds. And just like Valerio says, I do like the medium mixture, and I do 1 seed per container to find out what is the wildest and the looking one and the coolest one. I try to make one each year, which usually takes about a year for it to produce a callus and just to find out what it's going to make. Once we find out about that, then I start propagating and making more of that same one that has that unique look. I've done a lot of them, like I'm trying to think, like one of mine is toxic and destruction, and some of the seeds from Stephen Wang called the purple people eater, vortex, EEC vortex. I'm still in the learning process of tissue culture. There's a lot of information that's out there, steps, procedures, chemicals, a lot of great guys out there that I search for help. I have a few names. One of them is called Frank Travone, big tissue culture guy. Great. He has a tissue culture group on Facebook. Another one is Steve Seranian. Big help. I used to do a lot of pressure cooking when I used to do tissue culture, which I moved away from all that. That's really time consuming. Moved to the plastic containers. from plant cell technology. There's a guy named Francisco, which helped me out a lot of that too. And they're in the US, which I also buy their ppm from, which is the chemical that helps stop bacteria and microbes in the containers. A lot of work.
So, Craig, let me ask you about your prickly pear. It has a lot of scaling on the petiole,
Yes.
and maybe Valerio also notices it in tissue culture. When they're really tiny little babies, you can identify that trait. I think Valerio was saying you can pretty much identify anything except
Correct.
for size.
Right. You can see some of the mutation. I use a magnifying glass. I have to look through the jar and find that very unique one. And then, you know, I'll get like maybe, you know, a handful of seeds, 10, 15 seeds, and I have like maybe 50 containers just to find that very unique one. Once I find that one, then I pull him out, then I propagate that one. And then I let that grow. And then, you know, of course, I got 15, 25 containers of the same thing. So, but that's how I do it.
All right. So now
A
we're
lot
going
of
to
the
take.
ones,
Go ahead,
a lot
Craig.
of the ones that are done by seeds are basically you can name.
All right, so we're going to take a little video from David to see if you guys are experiencing the same problems.
I'm David from Carnivoria, Czech Republic. I try to cultivate all groups of carnivorous plants, Dionea also, perhaps to inspire for your roundtable discussion. I can tell you some my difficulties and observations briefly. For example, the plant produces deformed flowers, completely whole flower. Pollination is not possible. Plants produce flowers with the absence of sexual organs. Flowers have stigma and stamens, but these are not fully developed. So pollination is not possible also. Stamens do not produce pollen. The stamens can produce pollen, but it is unable to germinate on the stigma. Even after successful pollination, no embryo is formed into the seeds, or seeds do not germinate. It's possible. It's possible. Failure to show the expected traits in the offspring, or sometimes wide variability of the offspring.
All right, so I hope you guys all were able to hear that. He also sent me a little email. It's interesting. Some clones like Maier, wacky traps, develop strongly reduced flowers, and I've pollinated them for the last 5 seasons and only got seeds once. It didn't germinate. In contrast, highly mutated clones like Master of Disaster and Maier, they produce full-fledged flowers even in the next generation, like Hanukkah's, butterfly, Supe, and they are, you can pollinate them and they produce germinating seeds. The offspring of Master of Disaster times Maier and Maier times Master of Disaster are variable, but with stable characteristics. All right, so Matze, same problems, or how do you overcome the problems when you get all these really misformed flowers?
Yeah, I know exactly what David is talking about. And I know David very well. So he's also a great flytrap breeder and has some very nice clones out. for example, the cross T is a clone. I think many people know that one. And this is one of the clones which has also a deformed flower. And it's very hard to get seeds out of it. So I try to pollinate it quite often, but they are very rarely making seeds. Or if you use the pollen and put it on another fly trap, it's a very low percentage that you get out of seeds. But I think there's not really something that you can do for it or on it. It's just a genetic mutation, and the genetic for sex is probably deformed. So there's nothing you can do. Or Valerio, do you have any suggestions?
Oh, I am of the same idea, Matze. If something is not possible, we simply need to use what is possible. So
yeah.
I believe that I completely agree with your observation. And as a tissue culture lab, we breed what is possible from seeds, and what is not possible, we use tissue culture, and we find some mutation using tissue mutation. I have some suggestion for the discussion, because we have done this year some very interesting research within the University of Padova, a well-known university here in Italy, because we have tried to identifying some genetic variation on Venus flytrap using molecular markers. We have tried to extract DNA and find the right molecular markers to identify a variation. And we have found some very interesting information, researching both very distant, genetically distant, or at least very far away, very distant looking cultivar, like, for example, a giant all red or a microdent all green. So very different looking cultivars and very similar looking cultivars. So we have found some very interesting information about that. For example, the all red pattern is a very small mutation that generate the all red pattern in a cultivar. So, for example, using molecular markers is quite difficult to identify the differences between the all red, for example, periscope from red periscope. It looks genetically almost identical, almost similar. While, for example, the crested pattern of the Schapenstiel is quite market feature, and I believe that everyone that has some experience with Schapenstiel breeding has seen that it remains in a lot of seedling generated from Schapenstiel. So, for example, we have seen that the Schapenstiel feature is very distant from encrested petiole, is very distant from standard petiole, so is a strong feature. And we have seen that is quite recognizable with molecular marker. And that's to say that there are some feature, for example, that is quite easy to keep also with breeding, because they are strong pattern that maybe is also not recessive, is quite dominant in the genetic of some line. Some other that is very, very small, for example, the all red feature, and maybe it's quite more easy to obtain from a single mutation of the zone that characterize that feature, that all red pattern, then and it's very difficult to obtain using traditional breeding, because the genetic pieces that allow the expression of that feature is maybe very, very small. And so this is something that we have done, for example, to characterize the white mouth and to find we can call a pedigree to identify the very rare cultivar. We have started this project with this goal, and we are obtaining some very interesting results at the moment. Another thing that is very interesting I would like to discuss with Craig and Matze is the white variegated Venus flytrap, because for us it's almost impossible to keep the feature using standard tissue culture technique. So starting from flower stalk or from leaves cuttings, the white feature almost completely disappear in all our baby plants that is regenerating from cuttings or flower stalk cuttings. And while we have seen with traditional propagation from very simple multiplication from rhizome, will keep the feature, the white variegation feature. So I believe that is something that is not regulated in a single genetically. So a single cell is not able to manage white variegation, but is something that is due to a meristem, a growth point that is already formed inside the plants. And I would like to discuss with you if someone of you has some other experience than me regarding white variegation, because I have seen that pom-pom form and white variegation form. In my case, I'm not able to keep that feature using regeneration from leaves or flower stalk. I'm able to keep them only using standard regeneration, leaving the rhizome multiply naturally, so without inducing any regeneration. And I would like to discuss with Craig also if he has some experience with it. Pom-pom form and white variegation flytrap is my trouble, my nightmare for to keep the features in vitro.
Yes. All right. So before you guys answer that, let's listen to a little message from David once again.
Traits instability, which can manifest even after 2 years of cultivation. For example, I have not tested the use of colchicine or X-rays effect. I wish you an interesting discussion and the best regards from Czech Republic. Bye.
All right. So that's exactly right. David also has stability issues. So Craig and Matze, tell us your problems about traits.
So well, I can tell a few things about pom-pom forms or also the white forms, because this is a mutation which can happen sometimes randomly, and it's just in a single leaf, but not in the genome of the plant. And then
Exactly.
it's just for 1 or 2 leaves, or it just happened randomly. It can happen with every flytrap because of some cultivation shocks. Let's say it like this: if the temperature drops down, if they have some pests on it or something, in a reaction of this, the plants can produce white leaves. And but this is just a randomly mutation which is not stable. And then we have to focus on the mutations which are in the genome, and they are stable, and this we can use for propagation too. But the white leaves or also the pom-pom forms, they are like seasonal, so they're not all during all the season with white traps or with pom-pom traps. So it's every time different. Some plants, so well, if we talk about explosion, for example, I talked about this plant in the beginning, which is a pom-pom form. I have like 50 plants next to each other, and some have in spring, they have a lot of pom-poms. When the other plants are looking normal, they just produce the normal traps. Then in summer, the other ones produce pom-poms, and the other from the spring, which have the pom-poms, produce then the normal traps. So it's always a seasonal thing, but it's not like that a pom-pom form produce only pom-pom traps. They always have 2 kind of traps, like normal traps and pom-pom traps. And the same is with the white plants. They also have like 2 types of traps, like the normal traps, which are in green or red coloration, and then the traps with, or the leaves with white coloration in it. And it's also a seasonal thing. About the propagation, well, we do this in by dividing only. I have no experience with tissue culture in this case, but if you do rhizome dividing, you have the mutation stable in the new plants too, because this is a mutation which is in the genome, and then when you divide it, it's stable there. Yeah, Or Kenny, do you have any ideas more?
Let's go to Craig first, and then I'll follow up with that.
Yeah.
Okay, on the pom-pom thing, I do have some in tissue culture right now. And like Matze was saying, that I do have some that grow like mad with the pom-poms, and then you look around in the container, like say on the backside, and there's nothing. But they came from the same plant, the mother plant. It is a seasonal type thing I've seen. They grow very slow. They're very they don't grow very tall. In the tissue culture situation, they take a long time to grow. It's also done with a lot of the chemicals. I don't use any IBA. I don't use any BAP, which is another growth hormone. Mostly all my plants are in 1/3 media, which is very, it's a simple complex of distilled water and sugar and MS and ppm and get your pH at the right level, usually like 5.6. And, I'll make like 100 containers of them. And
Craig, sorry, but have you started from seeds or from?
yes, I started some from seeds, which I do quite a few, which gets overwhelming with too many plant containers.
So the pom-pom form, have you started the pom-pom form from seeds,
yes,
right?
I have that one in tissue culture. Yes, I do.
Okay. That's the point. That's the point I would like to, because that the pom-pom form that he's able to keep its features also from seeds could
Yes,
be
I got that.
kept also
I got
in
that.
vitro.
Yes, I got that from like a group of 10 seeds. And of course, this one produced the pom-pom, which I kept that one and started dividing it. The other ones, I kind of let them go because they didn't show any signs of that.
So Craig, was it like cheerleader times something, or was it pom-pom times pom-pom?
It was oh gosh.
Or whatever the parents were. You started with 10 seeds, and
I think it was St. Patrick's beard with, gosh, I don't know what the other one was.
Maybe was it a red one, like St. Patrick's?
It might have been. Yeah, that might have been it.
So out of the 10 seeds or 20 seeds, one of them showed the pom-pom, or
Correct.
all of them? Okay.
Correct. And then use that one specific seed that grew into the plant and then started dividing it and making more out of that one.
All
now,
right.
Some of my other plants, go ahead. I'm sorry.
No, go ahead.
I do a lot of leaf pullings. I do a lot of leaf pullings on plants and try to, and cultivate that way.
So speaking of cultivars, all three of you have experience with this. As a consumer, should we be registering or naming pom-pom forms and variegated forms if they're not stable? And maybe I'll go back to Valerio. Are you saying that you have a university who does the entire genome sequence for each, the ones that you're sending, or they're just looking at a segment?
We are using some already registered Venus flytrap genome from other research projects, and we are building primer on that section. So yeah, we are not researching on all the entire genome of Venus flytrap, but we are amplifying very kept region inside the DNA. And so we are using primer designed on that region that are already registered. So we are not sequencing all the Venus flytrap genome. So I know that the result is statistical relevant, but not complete. But at the same way, we are collecting the very first results on in a very interesting, I believe, project. What I would like to focus regarding pom-pom and white form is that I believe that, yeah, there are some pom-pom forms that is able to keep its features on also from seeds, but most of them I know very well that they have two type of traps, the pom-pom traps and the standard one, like as well the white feature. They are not able to produce white leaves all year round. But what I see is that from, for example, white mamba, or I have tried, it has been since 3 years I'm testing for in vitro introduction of pom-pom forms and white variegated plants. I have white mamba, for example, that is producing since 3 years only white and green traps in greenhouse, but starting it from leaves cutting, so inducing tissue culture regeneration from single cell, it's not able to keep its features. So I'm saying that that cultivar maybe has some modification in transcriptional phase or in something that happens in the cells later that is not the genotype itself, that is able to be kept inside the Venus flytrap when we are talking about the standard rhizome division, as you said before, Matze. So using standard division also in vitro, if I am able to keep, for example, a white variegation in vitro, and I induce standard rhizome division also in tissue culture, the feature is kept. While if I detach the leaves and induce regeneration, the mutation will be lost. So what I'm saying is that maybe some pom-pom forms and some variegation white flytraps, also that the one that is considered stable, if the mutation is not at the genetic level, so it's not a single cell DNA that allow the production of this feature, but it's something that will happen later, maybe in the RNA phase or something like that, I don't know. Because it's kept when we divide the rhizome or we use already formed growth point, while inducing single cell regeneration is lost. So this is what I'm trying to focus in, because we have been researching on that since 3 years, and for some pom-pom forms we have tried a huge amount, also the stables one, to regenerate them from cuttings, and they don't capture the features. No. While also in vitro propagating, allowing that, as Craig said before, without using phytohormones, Venus flytrap tends at the same way to propagate from the base of the rhizome. Allowing flytraps propagating this way, the feature is kept also in tissue culture, but not with cuttings. So this is the point I would like to focus and to give a point of discussion altogether.
Yeah, I've done some cuttings on a, let's say the Kim Jong-un, and I've done like 10 cuttings of it and put it in tissue culture containers. Most of the ones have got contamination, and I still haven't gotten anything. They call it a strike. I had the same situation happen to my, it's called toxic. It has taken me over a year to make a strike out of that one. I've tried, and I could not get it to strike. You know, I get contamination, and then the culture is gone. Sometimes you can just take a leaf pulling and just stick it in spag, and it'll propagate that way, but it just takes forever, you know. Just to give you a general idea, as in tissue culture, if you were planting from a seed, it would probably take you over a year. Now, if you do tissue culture, it grows 5 times faster. It produces faster and propagates faster. And you have thousands of them. That's where it gets crazy.
I would like to answer to Kenny's question before, because I have talked about something else. Kenny asked about registration. I believe that yes. Oh. If only with standard propagation the feature is stable, also registering white forms and pom-pom forms has sense, of course. Leaves cutting propagation or something like that maybe should not be mandatory to for a cultivar registration, because there are some feature like this one that has kept on could be stable only using traditional rhizome division propagation, and I believe that it should be enough if the feature has been able to be stable for 2, 3, 4 years. Also, if the feature is not stable in tissue culture, so tissue culture is not mandatory for me, and the fact that the standard rhizome division is enough to keep the feature alive, I believe that is enough for giving a reason for a cultivar registration. So that's sorry if I am not answered before, Kenny, but this is my point of view regarding white variegation and pom-pom registration. Matze, what do you think about it?
Well, we are, yeah, we have, so if we created some new clones or select some new clones, we have them under observation also for like 2, 3 years to see if the mutation is stable. And first, if it's stable, then we start the propagation with this clone to make really sure that it's also a stable mutation, that is it worth to get a new name. And yeah, but but about the white forms and pom-pom forms, I cannot say how it works in tissue, but for us it works very well with the dividing rhizomes. And but maybe one interesting thing, so we have also some plants in one season, they don't show any mutation like a pom-pom, they just have the normal traps, but in the next season they have pom-poms again. So it's really up to the plant when they perform the pom-pom traps. But I know always the trouble with the customer, because they buy the plant and they have the pom-pom forms, and in the next season they don't have any pom-pom, and then they say, oh, it's not a real one, it's a fake one. And I say, no, keep calm, it's the real one.
Right.
But it's up to the plant when they perform the special traps. So it's very seasonal. And with the white ones, it's also like this. They are maybe a little bit harder. So for example, we have the white tiger. And this one is also, or the white zebra, green zebra. These are 2 white forms of us, and especially the green zebra is very randomly making the white leaves. The white tiger is more often producing the white leaves. Yeah, but it's really up to the plant, and I don't know which influences that it get the white color out. So this is a big mysterium.
Matze, you said that you're going to look at a plant for 2 to 3 years to make sure it's stable.
Yeah.
Before that, from age seed to 2 to 3 years, are you doing leaf pullings, or are you like looking at a single plant?
No.
Or are you propagating it?
Yeah. So no. So actually, we saw the seeds, and we can get a major plant in 1 year by the traditional way. So Flytraps actually working very good with us, and they grow very fast. So after 1 year, from one seed, after 1 year, we have a major plant with flower already. And then we just saw the seeds, replant them after like 3 months, and then they grow up, and we can select, or we can see where are the interesting ones, the interesting new forms. Then we select them out, put in a single pot, grow them in major size, and then we leave them in the pot for 2, 3 years to see if it's stable. I write on the label what's markable for this plant, what's special for this plant, so I can every year control if it still has this mutation or not. And if it has the mutation like 2, 3 years, then we start to propagate them. But in the meanwhile, the plant propagates by itself. So when we divide it after like 3 years, we have some smaller plants inside the pot already. And yeah, that's how we're doing it.
All right.
Kenny, maybe we should introduce a new way for the cultivar registration at the ICPS, because on the description it has, if I remember well, one of the definition is that plants should be propagated in a vegetative way by left cuttings. Maybe we should introduce a new description for white and pom-pom forms, saying that plants should be divided only with rhizome division, because also with leaves cutting or something else, the feature is not stable. So maybe this is something we can do to allow the registration of this special Venus flytrap.
If you take a plant and you do a leaf pull and it doesn't show the same trait as the parent, doesn't that imply that it's more environmental because it's not in the genome though?
Yeah, this is what I suppose. I have no answer at the moment. They are only questions. But
Yeah.
what I'm saying is that I completely agree with Matze, saying that also the variation is seasonal, but at the same way, using rhizome divisions, they are completely stable, and this is right, because my white mamba, for example, divided by itself in the pot, and I was able to generate some divisions, and yeah, they are very stable. But starting from cuttings, both from leaves and from flower stalks, and with cuttings, I mean that a cell that is inside a tissue lose their tissue role, and it become a embryogenic cell that a single cell is able to regenerate a whole plant.
Mm-hmm.
So in that, when I'm talking about somatic embryogenesis inside a cell, they are what we call cuttings. So the regeneration of a single plant from a cell. In that way, if the mutation will be stable in a genetical way, it should be kept also in a single cell regeneration. So that's why I'm saying that this feature is not genetical, but is something that happens later in more mature tissue that is already differentiated in different role. Every cell has their role. And this is why I'm saying that maybe that mutation is not genetical, but is something that happens after. So in an RNA stage or something like that, maybe methylation, I don't know, but something different from the single DNA gene.
All right.
This is my point of view, of course.
Yes, I like it. All right, we do have a couple of questions. One is from Alex Malikov. He says, or she says, what makes a unique cultivar? How do you know when your offspring is different enough to be worthy of registering or naming? And of course, this is an opinion. So, Craig,
Yes.
we'll start with you. When you look at a plant, so in the US, we kind of have a list of about 130 cultivars, but in Europe, there's a lot of unnamed cultivars, and like Jessica Poveda in Florida and Matze, and lots of people have like 300 different types of cultivars. So first off, how do you know that your plant is not, that the phenotype isn't really the same and you're just renaming something that already exists? Or
when I do like typically a seed, I'll get like the mother name and what it was crossed with, and then I'll go ahead and maybe get like 10 seeds, and I'll go ahead and tissue culture them. Since they do grow a lot faster, what I'll do is I'll monitor them and look for any weird or anything deformed about them. And when I do, I particularly take that one, and then I go ahead and propagate it to make more of the same one that was that weird type texture. The others, I just kind of like push them to the side. As I start to grow, let's say it's going to at least take me more than a year to find out the weirdness of it, the features, the teeth, the color, the petal, the size. Once it gets to that unique look, then you can name it. anything from a seed is not true to the name of the mother plant. Now, if you take the mother plant and you do a leaf pulling of it and tissue culture it, then that's true to the name. And then I take it and find a unique name, or and then write up a form of it, of the unique features of it, and what it was crossed with, and then I submit it, and then I keep it as that.
Valerio, how do you discriminate between something that you've just bred or something that's worthy of duplication and spreading?
Of course, this is something that is quite subjective, so it depends a lot on the equilibrium that everyone has in his mind. So if I know about all the cultivar that is in the world, I will have a quite clear idea of what is
unique,
so nice to be registered and what is not. Of course, I believe that at the beginning of everyone's career, we have something that maybe we would like to register only for a heart reason, because we love it. Maybe it was our first breeding, our first experiment with Venus flytrap breeding. So it's something that is subjective and objective in the same way. I believe that a professional company and a professional way of thinking is to mix the two things. So maybe one way to discriminate between something that worth a name and something that is a simple Venus flytrap seedling is maybe to start and showing it publicly in social network and looking at the response of the public. If there is a quite nice response from the public, it means that almost and also the request is quite high. It means that it's something quite new, or that it's worth to be registered. At the same way, but I want to focus is that there will be always a cultivar that will be similar to another, I believe. So it is tolerable also to mix some passion inside the breeding perfection, because we are also human and we have some maybe cultivar that we love, and to report them in our website as the long name, the complete cross, is something that is not okay for a marketing reason.
Mm-hmm.
And so something that we love to name maybe 1, 2, 3 cultivar that we love because they were our first. It could be tolerable also if they are very similar to another ones. But when we are talking about huge number, I believe that the market response is the most important feature to keep in eye and to underline when we should register something. So I believe that we should start showing a Venus flytrap 1-2 years before naming, to wait for the public response and then to decide if it is working a name or not. This is my point of view.
That's a great reply, because I remember when Craig posted definitely prickly pickle, but maybe some other things, and it's getting like 1,000 likes and a long list of people saying, when is this available? I'm ready.
Yes, I get pressure.
So Matze, I know in the US, and I'm sure Europe, people are very familiar with your cultivars, with their mutatedness, with their size, and you're saying you wait 2 to 3 years. Do you have siblings of these registered cultivars that didn't make the cut?
Maybe first I want to say thank you, Alex, for this great question before. And I can go with both opinion from Valerio and from Fred. And every single seedling plant is genetically different from all other plants, so it's an own clone. But as Valerio also says, it makes no sense to name every single plant. And if we go, for example, with the dentate forms, if you sow the seeds from dentate traps, you get out a lot of dentate traps, but it makes no sense to name them all. But coming back to your question, Kenny, so it's difficult, especially with the giant forms. Let's take this for an example, because when we make the crossings with giant forms and then we sow the seeds, you have to grow them first like 2-3 years to get them in real major size to see if they produce very big traps. And there we have a lot of seedlings at it. But I keep them under observation to make sure that I select the biggest plants of it. And with crazy traps is different. So when you sow the seeds from a nice crossing, and let's take maybe a werewolf, for example, if you produce seeds from werewolf, you get out a lot of different forms, different crazy forms. Some are looking like werewolves, some looking more like a mirror or master of disaster, and this is mixed up with everything. So there it's very hard to select the very, very good ones, but we always try to find the plants which looks completely different from that what we have already. So it's always up to the breeder what you want to select or what you want to choose. And especially with werewolf, they're coming out so many very nice things, very ugly things. And yeah, and we try to focus on the very, very strange and crazy traps. I think Valerio do it the same thing. And yeah, then you have a lot of plants which are no bad plants, but they are not worth to keep it or to give a single name. So we are selling this on garden markets, flower shows, or
Unknowns.
sometimes yeah, unknowns.
That's how I sell them.
Yeah, that's true. Or sometimes they go to the compost because it's just too many.
no.
And we cannot keep them all.
All right, guys, we're almost out of time, but I have 2 more questions. But actually, I have 3 questions. One is for Matze. Are you using grow lights to get them from seed to?
No.
Nope, just normal.
No, just normal sunlight. So actually in our nursery, we keep everything very, very simple. We don't have much technique inside. We don't use grow lights, and just normal sunlight, a good soil, a little bit of fertilizer, and that's it.
All right, very good. So we got two last questions. One is from Brian Del Bianco. I would like to know if the cultivars have reduced cold weather tolerance when compared to typicals. I lost a red dragon and a B52 that were in my garage, but the typicals in the same condition seem to be fine. All right, so the 3 of you all talked about size and mutations and color. Is anyone breeding for temperature range or environmental range? Or we can also, we should answer Brian's question, of course. Do you find cultivars to be less stable in environments?
Hardy.
Yeah, hardy.
Can I answer for first, because I need to go in some minutes.
Yes, we're going to be done in 6 minutes.
Oh, okay. So yeah, we have selected. We are trying to select something that is highly seasonal, because as Matze said, we also exhibit our plants at different shows, and yeah, especially sending plants as typical, but not only typical. And really, we are talking about a lot of different variety. We are trying to select plants that is maybe more cold tolerant, and we are able to sell very nice looking plants also till late October, late November, because yeah, there are some cultivar that suffer maybe a lot the warm weather in summer, but then is able to keep a very nice green and open trap also till the beginning of December, for example. And we are simply selecting by looking at different cultivar behavior during the different season. And yeah, there are a lot of interest into selecting very nice Venus flytrap for the general market that is maybe very nice looking at the beginning of the spring or at the end of the autumn or in the middle of the summer. That is the season in which plants tend to forms maybe brown leaves or something that is not so beautiful to see. So we have something, some selection also based on the best season of each plant, because when we are going to sell typical plants to the general market, we select in spring the spring variety, in autumn the most autumnal variety. Regarding the question of Brian, I don't believe that there are different hardiness regarding typical or selection, but hardiness doesn't matter on name of the plants. It's something that it depends only of genetics. So there are genetic mutation that generate very weak plant, like for example the very weird mutated plants. I believe that a lot of werewolf seedling is very low heat tolerant, and it has a very it suffers a lot the very warm weather in summer, for example, but it doesn't depend on the fact that it is typical or not. It depends on genetics. So usually where the flytraps are more weak than the standard one. So from this point of view, statistically, the typical flytraps are, of course, it seems to be more vigorous than the mutated one. So this is something true. But at the same way, there are a lot of cultivar and different Venus flytrap names and also famous name that is very vigorous as a typical one. So this is what I think about it. We are doing some selection, not only based on forms and size of traps, but also on the seasonal best of each variety, which is the best season for each one. And the typical is not enough to say that is more vigorous than another one. It depends of the genetic identity of the plants.
All right, very good. So we're going to do a quick fire round. This is the last question. Can each of you name a cultivar or grower, probably cultivar, that you like, that you did not create? Some cultivar that we might be familiar with. Valerio, do you have a cultivar in mind that you really like, that you maybe wish you had invented?
I have one that is made by one of us at the moment, that is the bloody nurse. I have never grow it, and I would like to have as soon as possible.
I should send you some then.
What was the name of it?
Bloody nurse, the bloody nurse.
Okay, bloody nurse. Yes. Yeah. All right, Craig, what's one cultivar that you really like?
One of the best ones I like is called the GJ Cujo.
Ah, very good.
Yes,
the giant kudo. Yeah,
those are good. Those are cool.
All right.
Yeah, that's a nice one. It's a great honor for me that you all like my clones and want to get them the most.
we don't have them in the
so
States
well for me.
I know, Matze, you cannot name yourself. So
no.
can you?
No. Yeah, there was a clone. I had it once, but sadly I lost it. And this was a red pom-pom form. And it came up randomly of a friend of mine by his seeds. And he gave me a small division, but it doesn't survive. And I don't think that it's in cultivation anymore because my friend lost it too. But this is a plant where I really want to get my hands on it, or like a red pom-pom form.
Yes, and so do we. All right, so thank you, gentlemen, so much for participating in our roundtable. And for all those listeners out there, we encourage you to breed your Venus flytraps and to share what you create. And hopefully you can create something cool like these guys did. And we're looking for a red pom-pom. All right. And with that, thank you, everyone.
Thank you very much.
Thanks, guys.
Matze is going to go to bed because it's 1:30 a.m. Bye, everyone.
Vietnam here, yeah.
Bye-bye.
Bye-bye.
Bye, thank you.
Thanks for
Bye,
watching.
guys.
Thanks for watching, guys.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.